All right, we're finishing up our talk on genetics from last week. So let's start right in with epistasis. So epistasis is the expression of a gene at one locus that alters the expression at another locus. So um, two genes on two completely separate chromosomes, or maybe on the same chromosome but in different locations, affecting one another. And we see this a lot of times with a lot of different genes. So most genes, or most characteristics, have multiple genes that control the feature. So this is one clear example of this. In Labrador Retrievers, we have black and brown pigment for big B is dominant for black, and little b is recessive for brown. And we also have another gene that controls for having pigmentation at all or having no pigmentation, and that will produce yellow labs, which we'll see in a minute. So for this thought experiment, we have a heterozygote black lab being mated with another heterozygote black mat, so black lab. So both of these are big B little b and big E little e. So both are heterozygotes, and we would expect a ratio of 9:3:3:1, but what we see is 9:3:4, and that is because we have this double recessive hiding what would normally be black labs here. So notice the little b little b's, little b little b, little b little b. Normally, this would be another brown coat, little b little b. This would be black lab, big B little b, big B little b, big B little b. All three of these would be black labs. But instead what we see is 4 with this other trait, which is the double E recessive. So this double E recessive is showing up as this 4, and we have 9 and 3 of our other phenotypes. Notice how if we separate out these three, these two heterozygotes and this homozygote dominant, we would get the normal 9:3:3:1. However, when we have this effect of epistasis, sometimes this ratio will be slightly altered in that some of these categories might be added together. Depending on how that works, we can get multiple different ratios, but all of them will add up to 9:3:3:1 if we are tracking two alleles for a given cross. All right, polygenic inheritance. So a lot of different characteristics are the effect of multiple genes. So in the past, we've thought of things like height as being like I got the tall gene or I got the short gene. But really what's happening there is there's over 180 different genes that affect height in humans. So most outward characteristics that we like to think of, or at least many of them, are the result of polygenic inheritance. They have more than one allele that control them, more than one gene, more than one protein control that one trait. Skin color is another great example of this because you can sort of see the shades in between. And skin color is the result of, in this example, we have like an 8 allele cross. And with that, we can have a wide variety of skin tones. So based on the inheritance of different alleles, you might have a different skin color. This accounts for why in specifically in mixed race couples, why we can see variety between the children, because a lot of these alleles can end up in different being assorted differently between the two children. Because there's a random mixing between parents, one child might have darker skin and one child might have lighter skin, just depending on which alleles assort which way during meiosis. All right. So we've talked about the genetic impact of phenotype, but it's also important to take into account the environmental impact on phenotype. So um, I think when we're studying genetics, we tend to favor the idea that genetics control everything, but it's very important to keep in mind that the environment is going to have a large impact on on the outward appearance of an organism as well. Yeah, maybe not even the appearance, but the characteristics of that organism might also be affected. When we do have multiple impacts, we call it multifactorial. But I just want you to be comfortable with this concept, the idea that we can have things like hydrangeas, which if they are grown in a acidic soil with aluminum available, so with free aluminum, they will develop blue flowers. Whereas if they're grown in just a basic soil, they will grow out as pink. So a lot of what will happen with, and these are two in the same species, like this is the same variety. There's nothing genetically different between these two plants except the only thing that's different between them is the environmental differences. Um, and actually, that's one of the things we saw with we saw multifactorial development affecting height in humans, and that's one of the reasons why average height has gone up is because nutrition is much more readily available. So better nutrition, because better nutrition is widely available now, it is much more likely that children will grow up to be taller. Or in the past, when you've had uh, times where children weren't getting adequate calcium, weren't getting adequate nutrition on a very regular basis, average height was also lower because the human body will only grow as much as you give it. If you don't give the human body adequate nutrition, all sorts of deficiencies can develop, and one of those is during the period of growth, which is childhood. Um, the bones might not develop as much if you do not have adequate nutrition. Um, yeah, let's move on. So, an organism's phenotype includes its physical appearance, but also keep in mind those internal characteristics, so anatomy, physiology, and behavior as well. So, the large scale behaviors of an organism are attributable to its phenotype. And remember that behavioral things as well as genetic things can have an effect on all of these. So, behavior, physiology, anatomy, and appearance. So the phenotype is a reflection of the genotype, so all of the genes, as well as the unique environmental history, so the um, the effects the environment has had on the organism over its lifetime. And while we do see um, decent patterns for inheritance through Mendelian genetics, a lot of human traits don't follow this because they're multigenic, because we have, or polygenic, sorry. So because they have many genes attached to them that contribute to that outward characteristic, a lot of traits cannot be studied with Mendelian genetics. There are specific examples that can, however. One very good and easy example is certain diseases. Also, um, uh, a lot of diseases, because they have such an impact on humans' lives, they're well recorded. A lot of times there is family trees that sort of chart out these diseases. Um, it becomes a lot easier to track them. Other characteristics like color blindness, um, and we'll talk about in a minute, which is PTC tasting is another good single gene trait. Um, but yeah, unfortunately, because our generation times are so long, because we have so few offspring, and because it's unethical to do breeding experiments, um, we're sort of it's difficult to perform genetic experiments on humans. But fortunately, our understanding of other organisms has helped to enlighten us with regards to our own genetics. And there are still perfectly acceptable ways to study human genetics um, through sequencing and testing proteins, things like that. So a pedigree analysis is where we're looking at a family tree and trying to track the transfer of alleles across generations. So which genes, dominant or recessive, are going across generation gaps here. And here is our example. So we're looking at two traits. Now I believe Widow's Peak is actually polygenic, but for the purposes of this example, we'll consider it a single gene trait. So let's look at this first example. So Widow's Peak, we have two sisters here. One is either heterozygote for big W, little w, or homozygote, big W, big W. And let's look at this trait. We have, at the very top, we have someone who is big W, little w, who's showing the disease, and someone who's little w, little w. And when they have children, they end up with two that are homozygote recessive and two who are heterozygote. And those two that are heterozygotes show the trait Widow's Peak. We have two people who are heterozygotes who are showing that trait for Widow's Peak, who have children. So these are two different family trees coming together. And we have one child who has it and one child who doesn't. Now this is a result of a dominant trait. So this, specifically here, this is a dominant trait. We can see that because if we were to cross two people who had this trait, who are showing this trait. So if we had, let's ignore all the W's here. Let's have just this little tree here. If this was a recessive trait and we had two people showing the recessive trait, having children, all they would have is little letters to give. All they would have is little w, little w, little w, little w. And when they had kids, all their kids would have the same alleles then, little w and little w. So both children then would have the trait of Widow's Peak. So keep in mind that if you have two people showing the trait breeding together, having children together, then if you have any variety, then you know that they had to have been heterozygotes. At least one of them had to have been a heterozygote. And that's how we can sort of tell whether or not it's dominant or recessive. Let's look at PTC tasting now. So this is a chemical that tastes bitter to several people. You can get PTC tasting test strips. One of the things that we see with, specifically with PTC tasting, several vegetables taste incredibly bitter, like broccoli and Brussels sprouts will just have an incredibly bitter taste. And the trait to taste that is displayed here. Now, if we weren't looking at any of these letters, and we're just going off of having nothing here, and we were to have someone who doesn't have PTC tasting having children with another person who does not have PTC tasting, so both of these people are empty shapes, so they are not showing the trait. They have two children that do show the trait and two children who don't. Well, because that, we know they're heterozygotes, and if they're heterozygotes and not showing the trait, we know that trait has to be recessive. All right. We will see that 1-to-1 ratio pop up where we have two children to two children. That gives us a 1-to-1 ratio. That 1-to-1 ratio is highly indicative of a cross between two heterozygotes. All right. If you want to see the math behind that, you can do the cross between big T little t and big T little t yourself, and you will see that 50%, that 1-to-1. All right, and then when we look down at this next generation, again, we see a 1-to-1. So we have two heterozygotes crossing, two people who are carriers for this trait, who have one child that cannot taste PTC and another that can taste PTC. So this is a recessive trait. All right. And there will be an example problem at the end where we'll go through one of these pedigrees. So for that, I will give you the blank. You can try to fill it out on your own. Then I have an explanation video, and then later on I will give the answer after this is graded. So I have sort of a lead-in video specifically for pedigrees. All right, so pedigrees can be used to make predictions about future offspring. So based on pedigrees, you can predict the likelihood that children will have certain traits, and that's especially useful if you have a family history of certain diseases. So specifically a lot of diseases are inherited recessively. Um, and these can be anywhere from mild to life-threatening. So we have some mild genetic disorders like color blindness compared to life-threatening ones like cystic fibrosis. Red-green color blindness makes it so it's difficult to perceive the difference between red and green. And specifically, on the life-threatening side, cystic fibrosis is a genetic disease that causes the cilia in your lungs and esophagus, or your trachea, to be very weak and brittle, and that causes them to not be able to move mucus back out into your throat and then back down into your stomach. So mucus sort of builds up in your lungs, and that can cause bacteria to go rampant and cause disease. It causes all sorts of side effects, and it's not pleasant. It is an example of a recessive genetic disorder. Fortunately, with the advent of modern medicine, antibiotics have been improving as well. We have seen the life expectancy of cystic fibrosis patients jumping from about 30 years of age when I was younger, so about 20 years ago, to about anywhere from 45 to 50 years of age nowadays with modern antibiotics. So it is increasing drastically, and quality of life has also improved, although there is certainly many hurdles to overcome, even with modern medicine. All right. So when we look at recessive alleles, I referred to them earlier as carriers, heterozygotes. So if you have a recessive trait, and the heterozygote is sort of masking that trait, so it doesn't show unless you have two alleles for the trait. So in other words, it's recessive. Someone who is heterozygote will carry that disease. They are a carrier. And most of the times what you will have is you'll have a carrier parent or two carrier parents coming together, and that is when you see the recessive disease being expressed. So albinism is a perfect example of this. So in albinism, we have a lack of pigmentation in skin and hair. We see there's many other effects to the gene as well, but those are the most visibly apparent. So here we have two sisters again. One is showing albinism, one is not. And we see the the genotypes of both parents is big A, little a, and both of them have a normal phenotype. They do not show the traits for albinism. But when they come together, big A, little a, we do this cross. They have a 1 to 3, so a 1 in 4 chance, 1 to 3 ratio of children who get albinism. They have a 1 to 1 ratio, so 2 big A, little a's, and 2 not. So 1 to 1 ratio of being a carrier. And they have a 1 to 3 ratio of having a normal phenotype, so having a child that will lose the trait for albinism and will not carry that into future generations. So because big A, little a, because these heterozygote carriers have no decrease to fitness, they can have children just fine. Even in when we look away from human species, when we look out into animal populations where we do still see albinism, the reason why albinism still moves through the population is because it is a very small chance of creating a variant that would decrease fitness. So albinism, generally, when we look out at the effects of natural selection, will result in a loss of coloration. that will often confer a decrease in fitness, less likely to have children. But in human populations, we don't see that same thing happening. So we've sort of stepped the bounds of natural selection in that way, and that is more onto artificial and sexual selection. All right, so if a recessive allele that causes a disease is rare, it's unlikely that two carriers will meet. And especially if the recessive disease causes a decrease in fitness, it is much more likely, so those traits are selected out of the population, even within humans. One place where we do see an increase in genetic disorders, so where we see recessive alleles coming together more often is when two individuals are more closely related, so either cousins or second cousins, or get close enough between siblings or between parents and offspring. We do see a lot more times where these rare recessive alleles can line up, and that's because there's many different recessive alleles that can cause problems, but most of the times they're sort of spread out amongst populations enough to where they don't cause problems. But the closer two individuals are in relation to one another, the more likely those rare alleles are to meet up. That's part of the reason why lots of societies and cultures have developed taboos against close relative relationships, is because the offspring of those relationships were generally very sickly, very poor of health, and more likely to inherit genetic disease. And that is all because of genetic diseases. So as I said earlier, cystic fibrosis, what happens there is essentially the cilia become disrupted. So specifically, cystic fibrosis is because of the absence of these chloride channels. What happens is it causes those cilia to sort of break apart. They can't move. They can't lubricate that mucus and keep it away from the cilia, and they just sort of get stuck. So it also causes issues with absorption in the small intestine. I forgot about that as well. So it's lungs and small intestine. So they have nutritional issues that they have to overcome, as well as the issues of buildups of mucus. So that is an example of a recessive trait. So you have to have two broken copies of that. So two broken copies of the protein, or of the allele that codes for the protein for this chloride channel. All right. And we see also sickle cell disease. Now, one of the reasons why we see a higher incidence of sickle cell anemia, sickle cell disease, in African Americans is because of, believe it or not, natural selection. So because sickle cell, so if we have double recessive for sickle cell anemia, we have abnormal hemoglobin, abnormal cell shape, and all the hemoglobin is abnormal as well. So double recessive is where we see the actual sickle cell. That causes problems with clotting, so higher risk of stroke and heart attack, higher risk of blood clots in general. It's a very rough disease to have. A lot of times you have to treat it with clotting factors, or not clotting factors, like blood thinners, things like that, to help keep platelets from sticking together, help prevent clots from occurring within the bloodstream. However, one thing we do see with sickle cell is that heterozygotes are less susceptible to malaria. So even if you have you have one normal allele, one sickle cell allele, you are more resistant to malaria. So if you are homozygote dominant in this case, so homozygote recessive would show sickle cell. If you are at homozygote dominant, you're more likely to get malaria. So between those two extremes of either dealing with a higher chance of malaria or dealing with a higher chance of sickle cell, heterozygotes are the ones with the advantage there, and that has led to a higher population of heterozygotes within Africa, or within the African populations that descended from Africa, um, and that has led to a higher incidences of this allele, and that's one of the reasons why sickle cell is more common in that population, is because there's a selective advantage to being a heterozygote if you live in an area that has malaria. Malaria is a mosquito-transmitted disease. Fortunately, we do not have that in the U.S. But yeah, that's also one that is very many foundations are working on a vaccine for, because malaria is a truly awful disease. It's one of the most lethal organisms for humans is mosquitoes, because they transmit diseases. One of the most deadly diseases that they transmit is malaria. And fortunately, it's only certain species of mosquitoes that transmit malaria. All right. We do see some dominantly inherited disorders. They're generally more rare, but they do occur. Um, because a lot of times they're a result of a mutation, so they can be passed down, but a lot of times they don't. They have rather sudden origins in within the scope of like a family tree, where it is a break in a specific allele that occurs and then can then be passed down. But a lot of times in the past, these have conferred a large decrease in fitness. Um, but one example of this resulting in a trait is dwarfism. So we have someone who has the allele for dwarfism. They're a heterozygote for it. They are showing that phenotype. They are showing the dwarf phenotype. And they are having an offspring with someone who does not have an allele for that. 50% of their children will show the phenotype for dwarfism, and 50% will not, because 50% have a chance of getting the dwarf allele from the parent, or from the single parent. Because if we look at the Punnett square here, big D goes down here, little d goes down here, and obviously the mother in this example only has little d's to give. We end up with that 1-to-1 ratio. All right. Timing of the onset of a disease will affect its inheritance. So if a disease expresses itself after reproductive age, so for us, that's generally after 35 to 40 years of age. Most people do not have children past that. Obviously, exceptions do occur. But the later in life those diseases present themselves, the more likely that they are to sort of sneak through the population, because a disease will only decrease an organism's fitness, its likelihood to have children, if it occurs while they are still reproductively active. So because a lot of these diseases happen after an organism has stopped being reproductively active, there's no selection pressure against the disease. There's no decrease in fitness that will decrease the frequency of those alleles. So we have one example of this is Huntington's. So after 35 to 40 years of age, Huntington's will start to cause deterioration of the nervous system. It's nasty disease. There's no current treatment for it, or at best we can treat symptoms and help with quality of life, but we don't have any treatments to treat the disease itself. And another thing we can think about too is other things that have multiple factors that impact them, so multifactorial disorders. So things like heart disease, cancer, alcoholism, and mental illness um, can all have genetic as well as environmental components. Um, so whether or not you're exposed to, let's say, a diet that is conducive to heart disease, or a diet that's conducive to cancer, or perhaps a job that's conducive to cancer. So an example there would be people who mine coal will have a higher incident of lung cancer. And that is not because of their genetics, that's because of their environment. Alcoholism, another example of that, where someone might be a higher likelihood for alcoholism, but perhaps they live in a culture that abstains from alcohol, maybe a religious, if not a, sorry, if not a like a local area thing, a cultural thing. Another example is mental illness. So a lot of times we can have environmental figures or environmental factors trigger mental illness, um, but we can also have genetic factors that increase or decrease the likelihood for mental illness. One of the things that we're starting to see now is genetic testing and counseling. So people who have a family history of certain diseases might seek out a genetic counselor before they have children. And then the counselor can sort of tell them the likelihood that their child will have certain genetic diseases. Specifically, like if you have a family history, like if you have a sister or um, a parent or relative that has cystic fibrosis, and your partner also has family or relatives that have cystic fibrosis, it definitely would benefit the two of you to look into a genetic counselor to see what likelihood you have to give or to have a child who has cystic fibrosis. Um, and obviously, we're human beings. We can't tell people no or yes, you can or cannot have children. It's unethical, but they can at least give advice on and let them know the likelihood of a child contracting these diseases. And aside from looking at family history and going through pedigrees, genetic counselors, with the advent of better gene mapping, so figuring out which genes go to which diseases, and more affordable genetic analysis, it's much more possible to look at the actual genes of the parents rather than just looking at pedigrees, which is an interesting revolution in that field. All right. So with this example, we're looking at a couple both having a sibling who died of recessively inherited disease. So they would know, they can make guesses, excuse me, based on both of the parents' pedigrees, what chance they have of being a carrier, and in fact, even better, we could look at the genetics most likely of a recessively inherited disease and figure out whether or not they are a carrier. Um, but if they are both carriers, then they have a 1 in 4 chance of giving the child the disease, as we saw with the albinism pedigree, where you have two big letter, little letters crossing, two heterozygotes crossing. There's a 1 in 4 chance that the child will end up with two recessive alleles and show the characteristics of that disease. They will actually experience that disease with the double recessive. All right. There is also a number of tests that can be done on fetuses um, to determine whether or not they will experience given diseases. So you can sample fluid from the amniotic fluid or from the villi and the cervix, and both of those will allow you to karyotype to get the genetics of the child. Once you have the child's DNA, you can determine whether or not it will have certain genetic traits. So one common test is for PKU, recessively inherited disorder that occurs fairly frequently, at least one in every 10,000 births. And here we have our first question. So this is on page 293 of the textbook. It's question number 13. Remember, when we see colored in shapes, we know that they are showing the trait. When we have empty shapes, they are not showing the trait. When we have squares, they are male. When we have circles, they are female. So do your best to track this disease. Does it appear dominant or recessive? Do your best to answer that on your own. Label out the phenotypes and genotypes, or label out the phenotypes specifically, and then do your best to label out as many genotypes as possible. So I want you guys to look at this problem, look at the image, and try to fill this out as best you can without looking at the video. Then look at the video and work through and try to figure out how the system works. All right, and goals for next class. So next week we'll be beginning chapter 16, looking into DNA and RNA. So with that, I hope you have a wonderful weekend, and I will see you all later.